For our research, we aim to explore the relationship between supplemented creatine and the resting memory potential of crayfish muscular tissue.
This research is novel for two reasons. Firstly, most studies with creatine explore the systemic effects of creatine on muscle, such as power and endurance. Second, other studies show the effects of creatine in other parts of the body, such as the brain and heart. We focus on the molecular mechanisms in skeletal muscle.
This graph shows an action potential. Action potentials occur in neurons, and they communicate with each other to allow the body to perform specific actions. They're also required for muscular contraction. When an action potential occurs, the charge of the cell becomes more positive, and that boosts it above the threshold and allows these events to occur, allowing the neurons to communicate with each other, which communicate with muscle cells, allowing that contraction. These action potentials require stimulus. In this case, we believe that the stimulus will be creatine. So for our hypothesis, we think that adding creatine to the extracellular solution of crayfish muscle tissue will depolarize the cell, making it more positive. We used a fiber optic illuminator and a dissecting microscope to perform the dissection.
The first tool that was used was the large scissors, which allowed us to cut off the head, which allowed easy access for the muscle tissue. After that, we used the scalpel to cut the cuticle down the midline and parallel to the sternites, which I will explain later, to allow the muscle tissue to be exposed. And to help out, because my hands are way too big to pull the cuticle off, we had to use the forceps to make it easier to make those cuts with the help of the Vanis scissors. And then lastly, we had the crayfish in this sill guard dish, and we had to pin it down with the 4 pins so it wouldn't move around in the dish and make it easier for us to make the cuts and not make a mistake. To model the dissection, we have a picture of what the crayfish looks like, and of course we already cut off the head, so I'm not going to worry about that. For that, we did use the large scissors, as I mentioned earlier. So first things first, we had to use these scissors to cut off all the swimmerets, and the swimmerets are just the little parts, they're like the little legs that float off from the body. So I'll just cut these off. Now that those are out of the way, the next thing that we did, we focused on just one of the segments. We didn't want to go into cutting all of them because it would kill the crayfish a little bit too soon. So we just focused on this segment of dissection. So for this, we made one cut with the scalpel. We cut down the midline, which represents this blue line right here. And we also made one parallel to the sternites. The sternites are this little kind of cartilage section, and it's more difficult to cut through than the cuticle itself, but we want to make sure we leave those alone. So after we made the midline cut and then the parallel cut to the sternite, we then used the forceps so it's easier to pull the cuticle off and away from the crayfish, and then we can use the scissors again to make the clean cut to expose the muscle tissue.
In order to create our solutions, we use normal crayfish saline, and then we had approximately 5 grams of creatine monohydrates, and we used a scale to create that, and then we used 2 150 milliliter beakers, and then we use a volumetric pipette. We used SPSS 28 to perform an analysis on our data. We performed a correlation analysis and 2 independent samples t-tests. We used the ground pellet and put it into the Silgard dish for recording. And this is the micro manipulator. It would have a glass micro electrode. We used the coarse advanced knobs to adjust the micro manipulator and put it down into the crayfish muscle tissue. And then we proceeded to turn on the DC intracellular amplifier. And this is what we used to record. We did 3 different trials for each solution and recorded it into a table. This is a table where we logged all of our data before analysis. We have the 3 solutions. The first is, and the third are the control solutions. They had no creatine. The second is our creatine solution with 5.004 grams of creatine. We did 3 recordings for each solution, and based on the data that we did before the analysis, it appears that this solution's resting potential did increase in a positive direction slightly, and it did remain the same between the control solutions. This graph shows our correlation analysis. These 3 dots show our recording values from the first trial, which is with the control solution, These 3 dots show our values from the second trial, which was the creatine solution. And based on this, there is a positive correlation between extracellular creatine and resting membrane potential. However, this correlation is not statistically significant.
So this graph shows the difference between our trial with 0 grams of creatine or a control versus the one with creatine, which had about 5 grams of creatine in it. This shows that there was an increase of the mean resting potential. However, this increase was not statistically significant.
So this is our graph comparing the mean resting potential between our first and third trial. These were our controls, and there was no creatine in these solutions. This shows there was no statistical significant difference between our first and third trial, which means that our tissue was still viable and time was not a factor.
Because our results did not indicate a significant correlation between creatine concentration and increased resting membrane potential, we cannot technically accept our hypothesis. We have to reject it. However, there were many limitations in our experiment that leave us with lots of questions. So there are many limitations that affect our experiment. Past experiments have shown that these factors can affect creatine's efficacy in different organs. So these factors are age, dosage, fitness level, and disease.
Some of our limitations were we only used one subject, which made it difficult for us to actually have more data to analyze. The second limitation was our solution was too foggy because we added too much creatine, which made it difficult to visualize the actual busted tissue when we were reporting it.
And then lastly, previous literature has shown that creatine has a significant effect on the heart and the brain. So that makes us wonder why in our experiment, why there's no significance in our experiment.
Despite all its limitations, our research is still important and still contributes to the scientific field. And actually the limitations itself are what helped the experiment, because now that we've observed these limitations, we know what improvements we need to make when replicating our experiment and hopefully to improve it for next time.
Our research showed a positive trend, and we believe that we could see a significance if we included all these improvements that we know we can make and if the limitations decreased.
For further research, we could include more subjects, different types of subjects, we could lower the concentration, or we can do an extended period of time with creatine solution.
The cell has to become positive above a certain threshold, and then the cell boosts up and no, that's not right.
The first tool that we used was the large scissors to cut off the head of the crayfish to allow easier action to get into, access, wrong word.
Still viable, and their time was not a factor, and it shows that our usually after that.
Boop.
And then if I screwed up, then we'll just skip it.
Boop. Boop.